Hello and welcome to Alexander Disease Research Update, episode 25, recorded on January 22nd, 2025. I'm Albie Messing from the Weissman Center at the University of Wisconsin, and with me today is Charlie Wade from Queen Square Institute of Neurology at University College London, who's here to talk about his new publication on GFAP variants found in the UK population, along with Mel Feany from Brigham and Women's Hospital in Boston. Mel has been on before and is well known to all of you. But Charlie, since this is your first time on the podcast, can you say a bit about your background and current position?
Yeah, so thank you very much for having me on the podcast today. My name's Charlie. I am currently a PhD student at UCL. My PhD is on imaging of damaged white matter in both progressive multiple sclerosis and in adult-onset leukodystrophies. I'm also a neurology registrar, or as you say in America, resident. As you say, I work at UCL at the National Hospital for Neurology and Neurosurgery, which is in Queen Square in London. And I'm about halfway through my PhD and about halfway through my resident training, both of which, as I say, I work with a very good MS team and a very good leukodystrophy team as well.
Well, thanks again for being here. It's great to have you. Before we get started, please send feedback and questions to amessing@wisc.edu. So today our topic is a very interesting paper by Charlie and his colleagues entitled Analysis of GFAP Variants in UK Biobank Suggests Underdiagnosis or Incomplete Penetrance of Adult-Onset Alexander Disease, which is in press in the Journal of Neurology, Neurosurgery, and Psychiatry. And the reason this is interesting to me is that it addresses two big questions about Alexander disease. One is, how common is it? We typically call it rare or even ultra rare, but really we don't know. And the second is whether GFAP variants always cause disease, or whether some variants even cause problems other than Alexander disease. So, Charlie, let's begin by what you think is the main message about this paper.
So, really good question. My main take home is that these variants that can cause Alexander disease are more common than we thought. So they affect more people. But I think that there is a secondary point to be made, which is that if they are that common, then perhaps they aren't always disease causing. And I think, as I'm sure we'll talk about, there's sort of an understanding for a long time that maybe if you had these mutations, you definitely got Alexander disease. I think we've moved away from that slightly, but this paper probably contributes to that further by saying that actually probably not always, and maybe even less than most of the time. I don't know. So we'll sort of ask some questions that we still need answers to.
Well, let's start out with the UK Biobank. Many people are not familiar with that, so perhaps you could explain that to us.
So the Biobank is really an incredible resource, which was a prospective study launched in 2006 and recruited through till 2010. They recruited just over 500,000 people, and you had to go in for assessments. So the data that they received is incredible because at these assessments, sometimes you had a blood test, sometimes you had a cognitive test, sometimes they did questionnaires about your family history or your social history. So we've got lots of data there. Sometimes you had an MRI scan. And importantly, for this paper, just over 470,000 people had their genetic data taken. So they had what is called whole exome sequencing. So it's just a type of genetic testing. And so sort of we were able to extract a lot of the data from the UK Biobank and then look further into it, because as well as the data that patients actively submitted during those visits, they also linked to their NHS health record. So we could look at their GP or their primary care record and even their hospital record. So we could see not just the labeled diagnoses that those patients had, but also some of the symptoms that they complained of. So we were able to look at the Biobank, which, as I say, is an incredible resource. But then we were able to look across the Biobank just at the GFAP mutations we were interested in and look exactly what was happening to the people who carried them.
So we should say this is limited to people who were 18 years and older. Is that right?
Oh, good question. Actually, limited more than that, limited to people over 40,
Oh,
and only those through until I think 65. So it's quite a
oh.
small window of the population that you actually gathered, which does have implications for the way we look at the data afterwards.
But for those 40 to 65-year-olds, does this give you access to their medical records from childhood?
Yes, yeah, exactly. So it sort of gives you access to not just their medical records, but all of their data that they were happy to share. So sometimes we have their full education history, sometimes we have their full employment history throughout life. And then, as I say, sometimes we could get, if we could get I don't know, a record of their diagnoses, we could also find out when those diagnoses were made.
Okay, so I think you concentrated on variants that were designated as pathogenic, which means disease causing or likely pathogenic, based on computer algorithms that have been developed to try to analyze variants and predict their effects.
Yeah, so we looked for the first sort of filter we applied because you can't just look at 470,000 people, it's too much.
Right?
The first filter we said is just show us the very rare variants. So just show us the ones that are only occurring in 1 in 1,000 people. And then the next filter we applied were, as you say, the computational modeling that tells you whether or not something is sort of pathogenic or likely pathogenic. Overall, umbrella term was damaging.
But I think there are questions about how accurate those computer programs really are. I mean, that's one of the things that's really interesting about your studies because you find all these variants. Interpreting what they mean is a real challenge. How did you use these data to come up with an estimate of prevalence in the population?
So, I mean, I won't take any credit for this at all because my colleague Delia did it all, and she's the other joint first author. She's done this before on other studies, and I think she's done it very well here. Essentially, you look at the amount of, well, the carrier frequency, so the amount of people in the database who are carrying these sort of quote unquote damaging mutations. And then you factor in a number of other sort of variables into a model. And those variables include, and bear in mind, this is all based on UK population data. You look at the age of the UK and you sort of group them into different age pools. You look at the published, so the available knowledge that we have on so the age of onset of Alexander disease, and you look at how long someone usually lives when they've been diagnosed with Alexander disease. So you factor in a few different variables. You put it through a computational model, and then that sort of spits out at the end what we think a frequency would be, with some caveats, but what we think a frequency would be of carrying one of these damaging mutations. And as you say, the known knowledge of Alexander disease, that prevalence figure is variable in the literature. Sometimes, and I'm sure people who are listening to this podcast have seen, it's as high as 1 in 2 million, one in over 2 million, which means that realistically your chance of meeting another person with Alexander disease is incredibly slim. If you're in the UK, it might mean there's only 25 people in the UK with Alexander disease. In the States, it's probably higher than that. But with our prevalence modeling that we found, we thought that actually it was more like, as opposed to 1 in 2 million, more like 6 or 7 per 100,000, which would mean actually that in the UK alone, there were over 4,000 people with Alexander disease. So as opposed to there only being 25, we're saying actually there should be many more people, up to 4,000. And that was sort of based on the modeling that I've described.
So I think there are two really important issues to keep in mind here. One is that you filtered out the most severe variants because those would cause early onset in childhood and maybe relatively short lifespan. So they don't even make it to the age of 40. They don't appear in your variant list. The second is it seems to be equating having a GFAP variant with having Alexander disease.
Yeah,
I'm
so
not so comfortable with that. And
yeah,
Mel, I don't know if
I
you want
completely
to
agree.
chime in here too.
Well, there was one related question that I had for Charlie. which was, Albie just said, we filtered out the severe variants. And I wondered, Charlie, if you thought your data said that variants we consider to be severe, which you did not recover in your population, I wonder if that to you means these severe variants never cause milder disease. So do you think if the severe variants and that are severe in some patients that we know of had the capability of causing milder disease in other patients, would you have seen that in your data?
It's a really good question. So as we were saying, there is a sort of a natural filter to the biobank data because you had to make it to 40 and be well enough and sort of have the inclination to go along to an assessment centre. So there is a filter on patients who were too sick or too affected, or maybe just had too much else going on to take part in a sort of observational study. So we used, and as you say as well, we didn't find some of the variants that are very common in Alexander disease, but are normally seen, at least in the literature, in younger patient populations. And I think everyone interprets that as these cause a more severe disease phenotype, which presents at a younger age. And we didn't see any of those in this data set. I think it's interesting that we didn't see any of them. It might lend validity to the claim that they only cause severe and early onset disease. It might therefore not be surprising that we didn't see any of them. But I agree, it would add added strength to the argument that we think that, not always are these GFAP mutations disease causing. If we had found some of the more common variants, and we would have been able to expand our hypothesis further and say, even these variants that are associated with very severe onset sometimes are not always disease causing. We're not able to say that, from this data. I suppose from this data, you'd have to interpret it only as they're not present because they're so severe.
Let me come back to the title of your paper. It includes the word underdiagnosis and the phrase incomplete penetrance. Can you distinguish between those two? And I think a lot of people don't really understand what penetrance means, so that would be helpful to explain.
Yeah. So I think underdiagnosis is sort of one way of looking at this data. Underdiagnosis, sometimes misdiagnosis, sometimes because they simply haven't presented to their GP or their neurologist yet, would suggest that these people have clinical Alexander disease. So to go back to the beginning, they have the mutation that we think is damaging. They have clinical onset of Alexander disease and or radiological, so on the MRI, evidence of Alexander disease. And for whatever reason, they haven't been labeled as Alexander disease yet. Now, we're saying that it might suggest underdiagnosis, not because the patients weren't diagnosed in this cohort, because actually there's a sort of thing you should be aware of is that we actually don't know if these patients were diagnosed with Alexander disease. We didn't have an ICD-10 code for Alexander disease at the time, so it would not have made it through into this data set. But we're saying underdiagnosed here because we're not seeing 4,000 patients in the United Kingdom with an adult onset Alexander disease. We're the NHS specialist service. We should be aware of most, if not all, Alexander disease patients out there, and we're certainly not seeing 4,000. So I guess one interpretation of this data when we're saying, oh gosh, they're so common, these mutations, would be that actually these patients are being diagnosed as something else or are not being diagnosed at all. The other interpretation, and I think both have a lot of validity, and probably my concluding point will be that they both contribute here, is that mutations don't always cause disease. Now, that comes to the point of penetrance, and penetrance can be just really simply thought of as if you have 100% penetrant disease, that means if you carry the gene mutation that we know causes the disease, then you definitely go on to develop symptoms of the disease. You definitely go on to develop signs of the disease. And there are probably sort of increasingly few diseases that we think are 100% penetrant. We certainly used to think there are a lot more. But there are many diseases out there that people would have heard of, where if you have the mutated gene, you only have an increased chance of developing the disease. You don't always develop the disease. And those would be lower penetrant diseases. Now, certainly when I started reading about Alexander disease many, many years ago, my understanding at least was that if not 100%, it was very high 90s% penetrance for some of these mutations. And this data, I suppose, would suggest that actually possibly not. And that's not only because we're not seeing these patients in real life, we're not seeing 4,000 patients in the UK. It's because when we looked at the data of these patients who carry these damaging mutations, well, certainly some of them seem to have signs, symptoms, or radiological evidence of Alexander disease, but not all. And, this is a good data set. It's data collected over 15 years, really, now. You'd have thought that we would have seen if all of these mutations, which we think are damaging, if all of them were resulting in Alexander disease in the individual, you'd have thought that because the data set is so rich, we'd have seen evidence of that. But actually, we haven't really at all. So I think I couldn't give you exact numbers for either the miss or underdiagnosis rate or for the penetrance, but I think it suggests that there's possibly some element of misdiagnosis and some element of reduced penetrance that contributes to what we've seen here.
So I wondered, Charlie, if you can comment on what this underdiagnosis and reduced penetrance might mean for family members, particularly of patients with adult onset Alexander disease. So should they be seeing their neurologist or their primary care doctor bringing this up? Should they consider asking for genetic testing?
I think it's a good question because I think what we do with this data needs sort of quite careful interpretation. And I think I would have a conversation with your neurologist or your primary care physician if you are concerned of developing signs or symptoms of Alexander disease, especially if you have a family history of it. But I don't think that we're necessarily, with this data, such an early stage, recommending a change of practice. So I don't think that we need to expand on the genetic testing that has already taken place in England. I'm sure it's already taking place in the States. That, the predictor testing when there is signs or symptoms of the disease, I don't think we need to go further than that. And I am sort of very reluctant to say, as I always am, that people are misdiagnosing conditions in place of very rare genetic conditions. It's very easy in my position, where I see a lot of very rare genetic conditions, to think that other people should be doing the testing that we're doing more often, when actually, simply to get to our clinic, there have to have been a number of sort of filters in the way, excluding other much, much, much more common diseases. And so I don't think that I would, with this, be recommending a sort of significant change in practice and diagnosis. But I think it certainly is interesting. Alexander disease, especially adult onset Alexander disease, would not be one of the more possibly talked about, even leukodystrophies. And so we're now at a point, I think, where we could say maybe we should be talking about it, maybe we should be thinking about it. And, maybe especially when we're seeing some of the signs and symptoms we see or the imaging changes that we see, we should be pushing for genetic testing.
So let's talk about the question of whether any GFAP variants cause problems other than Alexander disease. Can you comment on that?
What we could get from this data, and I think it was just by the nature of the way we analysed it, is we thought, well, what we should be looking for in these patients who carry these damaging mutations is signs and symptoms that we know are associated with Alexander disease. And specifically, we filtered it a little bit more towards adult onset symptoms. So those with onset and younger age have slightly different symptom sort of pictures to the adults, who often have speech and swallowing problems. They often have sometimes breathing problems, sometimes bladder and bowel problems, all sort of symptoms that we might associate with the brainstem. And then maybe they have some memory problems and some behavioral changes as well, sort of early in their disease. And later on, some of them earlier in the disease can develop gait problems. So what we look for is, do the people who carry these damaging mutations develop those symptoms? And the answer was yes. So we found significantly so that if you carried a damaging mutation, you were more likely to have a bladder problem or a bowel problem, eye movement problem, or a speech problem. But we also found that they were more likely to have family history of other symptoms that we might associate with Alexander disease. So memory problems, you're more likely to have family history of memory problems if you carried one of these mutations. And we also found that they were more likely to have imaging abnormalities. So, which I should say would fit with Alexander disease as a diagnosis. So what we were looking for were signs and symptoms of Alexander disease caused by these mutations, and we found them. What we weren't doing was a slightly more, I guess, fishing expedition, looking for what else these mutations would be associated with, partly because I think that would be an enormous analysis, and partly because I think the statistics of it get a little bit more complicated when you're having to correct for so many variables. But we were only looking for symptoms, signs of Alexander's disease, and that is what we found.
Can I ask you, Charlie, if you, in this analysis you just described, tried to divide up the patients into patients where there's been well-described Alexander disease based on these mutations versus mutations that's really just your computer saying that they're damaging, to see if that would change your analysis at all and enrich perhaps for symptoms related to Alexander disease?
Yeah, so that's a very good question. As we said earlier, we divided them into we actually just grouped them all together really as a damaging, which is pathogenic and likely pathogenic. We found 36 mutations in 106 people. Now, 10 of those mutations had been reported previously. Something like 24 of them had been not reported previously themselves, so that mutation hadn't been reported previously, but another mutation in exactly the same location had been reported. And then in one of them, there was no other description of that mutation at all. So we had a good range. Unfortunately, when we tried to then further analyze, let's say, just the pathogenics that have been reported previously, or just the likely pathogenics that have been reported previously, the numbers became so small that any sort of meaningful analysis was impossible. And although I was sort of boasting about how rich the data set is, actually for each individual category that you go into, you realize, oh, not all 500,000 people had that test. So obviously some of them opted out. And actually of 500,000 only, quote unquote, 50,000 people had an MRI scan. And so we were left with our data set with only 6 MRI scans of patients who had mutations. So that gives you an idea of some of the times where we thought, oh, it'd be really interesting to look into just the published variants. But actually, when we looked into it, the data was so small, we were talking about 1 patient, 2 patients. So we couldn't, that's why we had to group them, I suppose. But yeah, another interesting caveat really is that not all of these mutations, in fact, most of them haven't been, described in an actual published case of Alexander disease.
So Charlie, how do we go from these predicted deleterious, predicted mutations that are supposed to be bad? How do we go from that to being sure they're bad?
Yeah, it's very interesting. So in these people, we can go no further. So for the patients in the UK Biobank, they are anonymized and always will be. So I suppose I'll never meet these people and find out, and I'll never be able to follow them up. In other cases, well, what we need people to do, as always, is publish cases that they see. I know there's a loop that people enter where a case gets returned to them as being a variant of uncertain significance, and therefore it won't be accepted for publication. But we sort of need to break that barrier, because one of the big sort of convincers, if you want, of pathogenicity sometimes is, has it been published before? And then we work very closely with our genetics lab when we do get variants returned of uncertain significance in GFAP and in other leukodystrophies, to think about tests of how we could prove pathogenicity and how we could then convince not just ourselves, but the sort of genetics community of the pathogenicity of a variant. The reason it's important isn't just for academic reasons. In the United Kingdom, I don't know about the States, but that's certainly how you unlock testing for the wider family. If you have a proven pathogenic variant, if you're left on the sort of VUS variant of uncertain significant status, you can go no further. So, it's a very important point that we sort of need to work on, is how can we, in real life, prove pathogenicity? In this data set, I think we've used the tools that are available that are widely accredited. So I don't know if there's sort of much more we could have done in the filtration process. And as I say, in terms of the next step for these patients, well, this is the end of the line for the data.
The other question is what really common variants in GFAP might be doing. And as you said before, you specifically focused on rare variants that were present at no more than 1 in 1,000. but there are some variants that are present in 5, 10, maybe 20% of the population. It'll be really interesting to see if they cause any problems, whether or not it's Alexander disease or something else. And as you know, a recent paper came out of a Japanese group describing D295N variant being associated with white matter lesions. So I think we'll talk about that in a future podcast, and maybe you'll come back and join us for that discussion, Charlie. All right, let's summarize. Charlie, what would you say again is the main take-home message from this study?
I think the main take-home would be that these damaging mutations in GFAP are not as rare as we previously thought. In fact, they're really quite common indeed, maybe affecting up to 4,000 people in the United Kingdom, which is not as big as the United States. And with that, we need to sort of think about whether or not they are all disease causing. So is the penetrance lower, which is a question that will be answered in future studies, I'm sure. Or are we not picking up Alexander disease as often as we should? So are we underdiagnosing it? And that makes us think a bit about our diagnostic methods. So I think there's probably more questions than answers from this, but I think that there's a sort of a good push now onto what other research questions we need to resolve.
Well, thanks again for coming today. That's all for today's episode of Alexander Disease Research Update. Thanks for listening, and thank you to Charlie and Mel for joining me today. Our theme song was written by Charlie Allenson, and thanks to our donors for these podcasts, the Barrett Riddle family. I'm Albie Messing. See you next time.